First of all, I would like to thank Thermo Fisher Scientific for giving me the opportunity to present my work on this platform. I am Ritu Kumar. I am Research Assistant Professor in the Department of Surgery at Weill, Cornell Medicine, New York. For the past couple of years, my work has revolved around understanding the epigenetic basis of pluripotency and epigenetic mechanisms involved in the reprogramming of somatic cells to induce pluripotent stem cells. In today's talk, I will mainly focus on an enzyme known as Activation-Induced Cytidine Deaminase, which I will call as AID, and its function in the establishment of naïve pluripotency. AID is known for its function in DNA demethylation process. Stem cells, as we all know, are very unique cells in the sense that they can self-renew, that is, that they can make more of their own, and when given the right signals, they can differentiate into specialized cells. Stem cells are present in the embryos as well as in the adults. Both adult and embryonic stem cells can self-renew, but the major difference between these cells lies in their potency. Embryonic stem cells have the potential to differentiate into all the cells of the germ layers, whereas the adult stem cells are less flexible and can differentiate into limited number of cell types. And therefore, embryonic stem cells are at the forefront of modeling and studying various diseases, as well as cell-based therapies for regenerative medicine. The pluripotent stem cells are present during the very early stages of embryonic development. Stem cells in the pre-implantation blastocyst constitute the naive state, which possesses unrestricted developmental potency and flexibility to produce all the lineages of the body. Conversely, the stem cells present in the post-implantation blastocyst are designated as epiblast stem cells and are less plastic and have acquired the genetic and epigenetic landscape for differentiation. That's why embryonic stem cells are derived from pre-implantation blastocysts. Contrary to mouse embryonic stem cells, which are similar to naive cells, human embryonic stem cells, despite their derivation from pre-implantation epiblast, are analogous to late-stage epiblast stem cells and are in prime state. This is a significant drawback for the translational application of stem cells, as prime cells are not at the ground state, are transcriptionally and epigenetically very heterogeneous, contributing to inefficient differentiation and significant variation and non-reproducibility within one and among various cell lines. Thus, recently, immense effort has been directed towards converting prime human embryonic stem cells to naive embryonic stem cells, largely based on knowledge gained from mouse studies. So, as also we are very well aware of the fact that pluripotent stem cells can be also reprogrammed from somatic cells, and Shinya Yamanaka have shown very elegantly shown that just by induction of 4 transcription factors OCT4, CMYC, KLF4, and SOX2, a somatic cell can be reprogrammed to a pluripotent stem cell, which is called as induced pluripotent stem cell. So induced pluripotent stem cells have immensely benefited the field of regenerative medicine and disease modeling, as now pluripotent stem cells can be derived from the somatic cells of patients, and these cells can then be utilized for generating various somatic tissues in a dish, which can then be used for cell therapy, drug testing, and drug therapy. My work started when I started looking for the function of the enzyme known as activation-induced cytidine deaminase in the process of somatic cell reprogramming. So AID is known to have function is DNA demethylation. So DNA methylation is the addition of methyl group to the 5' side to the 5' side to the 5 position of the cytosine, and it is the addition of methyl group is done by the enzymes DNA methyltransferases. DNA demethylation, on the other hand, is the removal of methyl group from the 5 position of the cytosine, and it can be achieved by passive and active mechanism. Passive mechanism is when the DNA replication takes place in the absence of DNA methyltransferases, and the active mechanism involves the function of enzymes such as deoxygenases, TET, and AID. So AID is a deaminase. It converts 5-methylcytosine to thiamine, and with the help of DNA-based oxygen repair system, the thiamine is replaced by cytosine, and that's how DNA is involved. AID is involved in DNA demethylation. So I studied the function of AID in the generation of induced pluripotent stem cells. And what I have reported that AID is required during the later stages of reprogramming, and it's required for the stabilization of pluripotency. So the generation of pluripotent stem cells from somatic cells is a relatively long process, and it can be divided into 3 overlapping steps: initiation, maturation, and stabilization. Initiation involves the first step is when there is a change in the cell morphology and the downregulation of the somatic program. Maturation is like upregulation of pluripotency genes, and stabilization involves massive epigenetic resetting, and this epigenetic resetting helps the cells to stabilize as pluripotent stem cells. So again, I have shown that AID is required at the later stages of reprogramming. Before I go into the details of my work, I would like to discuss a little bit about activation-induced cytidine deaminase. So as I have said before, that it is an inherent mutator, that it can convert methylcytosine to thiamine or unmethylated cytosine to uracil. The function of AID is known in B cell somatic hypermutation and immunoglobulin class switching and various B cell lymphomas. And it's a very well-known fact that activation-induced cytidine deaminase is outside immunoglobulin locus. So what I have shown, and we have published, that AID is required at the later stages of reprogramming. So what we see after 3 weeks of reprogramming using 4 Yamanaka transcription factors, that the AID knockout cells fail to maintain the expression of NANOG, as you can see in your left panel, whereas the wild-type cells express NANOG pretty homogeneously. And by 4 weeks of reprogramming, most of the AID knockout cells lose the expression of NANOG, whereas the wild-type cells maintain the expression of NANOG. And as you can see on the right panels, that we observe the similar pattern for other pluripotency genes, for example, OCT4 and SSEA1. And we saw that during the early stages of reprogramming, for example, 1 or 2 weeks, 1 week, that AID knockout cells have similar percentage of OCT4 and SSEA1 positive cells. But as the reprogramming processes, the AID knockout cells lose the expression of these 2 markers. So AID is required for the stabilization of induced pluripotent stem cells. However, what we have seen that we can stabilize these pluripotent stem cells either by continuous passaging. So when we pick up colonies, AID knockout colonies, after 2 weeks of reprogramming, when the colonies have not differentiated, and we continuously passage them, as you can see on your on the right panel, that if we continuously passage AID knockout cells, around 60% of the clones can be stabilized as pluripotent stem cells. Also, we have seen that when we play with the levels of transcription factors, for example, when I use lower levels of 4-Yamanaka transcription factors for reprogramming, AID knockout cells can be stabilized. As you can see on your left-hand side, when I use lower titers of lentiviruses expressing 4-Yamanaka factors, AID knockout cells can be stabilized as displayed by colony morphology and the expression of alkaline phosphatase. However, as you can see at higher levels, at higher viral titers, AID knockout cells fail to stabilize as pluripotent cells. So the question now we ask, the AID knockout cells, are they similar to wild type, or there are some differences? And indeed, we observe that AID knockout induced pluripotent stem cells are different from the wild type cells. As you can see on your left panel, when I looked at the expression of nanog at lower viral titers, I see that the AID knockout cells, they express nanog. However, the expression of nanog is lower than the wild type cells, and which is quantified on the upper right panel. And when we looked at the pluripotency expression of various other pluripotency genes, we saw the similar pattern that AID knockout cells have lower expression of pluripotency genes as compared to the wild type cells. And interestingly, we observed that these cells are primed for differentiation. When we keep them in culture for like 3 or 2 days, we see that there is upregulation of differentiation genes in the AID knockout cells as compared to the wild type. So this suggested that AID knockout cells are different from the wild type cells. So now that's why now we focused on if these cells if AID knockout cells are not naive cells, but they are similar to epiblast stem cells. So it has been shown by various groups that epiblast stem cells also have lower expression of pluripotency genes. They have higher expression of differentiation associated genes, and they are primed for differentiation. So, sorry, we were observing that AID knockout cells are more similar to epiblast stem cells as compared to the naive pluripotent stem cells, and that's why we wanted to study more. And it's a very well-known fact that the naive pluripotency is achieved in the presence of 2 inhibitors, MEK inhibitor and GSK3 beta inhibitor. Here I would like to point out the inhibition of FGF-ERK signaling using MEK inhibitor is very important for the maintenance of naive pluripotent stem cells, as it blocks the differentiation signals. Whereas epiblast stem cells are cultured in the presence of activin and basic FGF. So we decided to culture the wild type and AID knockout induced pluripotent stem cells in these media conditions. And we observed indeed, if you can see on your left-hand side, that in the presence of activin and FGF, which support epiblast stem cells, even after passage 1, AID knockout cells maintain the colony morphology, and they had homogeneous expression of nanog as compared to the wild type. However, when we culture these cells in the 2I media, which supports naive stem cells, we observe that the AID knockout cells fail to survive in these conditions. So AID, from these data, it indicates that AID is required for the achievement of naive pluripotency in vitro. So then we focused on the embryonic stem cells. So far, the data that I have shown you is after reprogramming somatic cells into induced pluripotent stem cells. To understand the function of AID in embryonic stem cells, we derived embryonic stem cells from the AID knockout blastocysts and started comparing those cells with the wild type cells. to see if these cells in vitro are also like epiblast stem cells. And similar to induced pluripotent stem cells, we observe that AID knockout cells have lower expression of pluripotency genes, which you can see on the left-hand side as compared to the wild type. And these cells were also primed for differentiation. If we keep them for 4 days in culture in the presence of 2I plus lip, they start to differentiate. So similar to induced pluripotent stem cells, it seems like mouse embryonic stem cells, AID knockout mouse embryonic stem cells, are also similar to epiblast stem cells. So again, to see if AID knockout embryonic stem cells can achieve naive state, we cultured them in the presence of 2I media, and we observed that similar to induced pluripotent stem cells, embryonic stem cells which are deficient, AID knockout embryonic stem cells cannot be stabilized in 2I media and they die. So AID is required for the establishment of mouse naive pluripotent stem cells. So we, in order to understand what AID is doing in these pluripotent stem cells, we performed, as I mentioned, that AID is involved in DNA demethylation. So we performed ERRBS to study the DNA methylation status of these cells. The data is on your left-hand side. As you can see that AID knockout cells are hypermethylated as compared to the wild type. And we could detect very many DMRs, and most of the DMRs were hypermethylated. DMRs are differentially methylated regions, and we observed that most of the DMRs in AID were hypermethylated as compared to the wild type cells. And another interesting thing we observed that in the AID knockout cells, there is hyperactive FGF-ERK signaling. And we believe that the AID knockout cells fail to achieve naive pluripotency because they have hyperactive FGF-ERK signaling, and it has something to do with DNA methylation. It's not clear to us right now, and further studies are required. So then we focused on, I'm going to share some of the preliminary data that we have for human embryonic stem cells, and I have mentioned that epiblast human embryonic stem cells that are cultured in vitro are like epiblast stem cells. So I use the simple I performed a simple experiment. So I created human embryonic AID-mutant human embryonic stem cells by CRISPR-Cas9 technology. So I picked one of the compound heterozygous clone for further studies. I targeted the exon 3 of AID, which codes for the catalytic domain of the AID, which has the deaminase activity, and started comparing this compound heterozygous with the wild type clone. And I observed that there was no significant difference in the morphology of compound heterozygous AID mutant and the wild type when they were cultured in the prime media. And as you can see that in the compound heterozygous, the expression of AID is low as compared to the wild type cells. However, very interestingly, I observed that these cells when cultured in the prime condition, which is very similar to the mouse cells, but we have observed that AID mutants are more homogeneously expressed nanog as compared to the wild type cells. And this was similar for other pluripotency genes, as you can see in the lower panel that AID mutant cells have higher expression of many of the pluripotency genes as compared to the wild type. So this indicates that in the prime conditions, similar to mouse cells, pluripotent cells, human pluripotent cells are more, they have higher expression of these pluripotency genes. And the question is, why do they are behaving like that? And for that, we looked at the FGF-ERK signaling. And what we saw that similar to mouse cells, uh, AID mutant embryonic stem cells have higher levels of phospho-ERK as compared to the wild types. So this could be one of the reasons that why AID knockout, AID mutant human embryonic stem cells are have higher expression of pluripotency markers. So the next question, the big question was if these cells can achieve naive state. So I cultured them in a media which is a commercially available media, RSET. Although there are many other kinds of media available now, but this is my first experiment. I performed these experiments in the RSET media. So as you can see in the RSET media, the prime cells, they are converted into they can be reverted into naive state. Just by looking at their dome-shaped morphology, which is a characteristic of the naive cells. And the naive cells, they have higher expression of the genes which are known to be associated with naive state, for example, KLS4, KLS17, TPX3, etc. It has been reported previously that when prime cells are reverted to naive, there is upregulation of some of the genes involved in the regulation of DNA demethylation, for example, DNMT3L and AID. I am interested in understanding the function of both of the genes in the establishment of naive pluripotency and to understand if they act as barrier or facilitator for establishment of human naive pluripotency. But today I will focus on AID only. And similar to what has been reported previously, I saw upregulation of AID in wild-type cells and prime cells were reverted to naive-like cells. And more interestingly, when I cultured AID mutant in this media, I observed that they don't look like the wild-type cells, as you can see on the left-hand side. The wild-type cells are more compact and they have more dome-shaped like colonies, whereas the compound head, which is a mutant for AID, has more flat colonies. And when I looked at the expression, which is the lower panel expression of various naive associated genes in the compound head cells, I saw that these cells, they fail to upregulate some of the genes that are associated with naive state. So this data suggests that AID is required for the achievement of naive state in human pluripotent stem cells also. Although this is I have very preliminary data and more work needs to be done for human embryonic stem cells, and of course I have to try various other media conditions to see if the cells behave the same. So in the end, I would like to thank a couple of people. First of all, Todd Evans, who is my mentor, and his suggestions and critics are very important for the study, and my lab members, and a former member of the lab, Jessica, who did the work on mouse embryonic stem cells, and our collaborators, Olivier Elemento, who has, which I have not discussed today, but he has done lots of epigenetic analysis on these cells, and Jayanta Chaudhary from Memorial Sloan Kettering, and we initiated this project in collaboration with him, and our new collaborator Thor, which is helping me in figuring out the conditions for culturing the naive pluripotent stem cells. And thank you very much for your attention.